Well, hello class. We're now going to look at the structure and replication of DNA. You've already studied this and you already know this, but we're going to review it just to make sure you remember it really well because we're going to go a lot more in depth. Just a quick review. Here's a person, right? Every person is made up of lots of cells, many, many cells. Here's one of her cells. Pull that out. Let's look at the cell. We know that there is an organelle called the nucleus, and inside that nucleus is what? A lot of chromosomal material, right? This chromosome, we took it out. If we take it out of the nucleus and look at it, here is the structure of it. We have two sister chromatids on that chromosome, and that chromosome is made up, if we start to unravel this chromosome, the chromatin that makes this up would unravel into DNA. So as we unravel this, there's actually very specific packaging that the DNA goes through in order to form that chromosome. But if we unravel all of it, we get these strands of double helix DNA. And you guys already know a lot about this DNA. We've studied it, but let's take a look at it again and remember what that looks like. So before we look at the specifics of DNA again, let's just step back for a minute and think about what DNA is like, how much of it you have in your body. You know that your body has a lot of cells. The different estimates, they're in the trillions. Every human being has trillions of cells in their body. Each human being also has 46 chromosomes in each of their cells, except for the gametes, right? Each of those chromosomes contains approximately 2 meters of DNA. So if you could take one of your cells out and take out one of the chromosomes out of the nucleus of that cell and unravel it and stretch it from end to end, it would be about 2 meters long. And within that 2 meters, there are approximately 3 billion subunits. So the ATCG bases, there's about 3 million base pairs in there, okay? Approximately 30,000 of those genes code for proteins that perform most of our life functions. Now let's do a little bit of math because this is really interesting. Let's take that 2 meters of DNA out of that one single cell, okay? And let's say the average number, there's different approximations, but the average number of cells in your body is about 3 trillion. So let's take those 3 trillion cells, multiply by 2 meters each. That gives us 6 trillion meters of DNA. If we would unravel it, lay it all out end to end, it's real skinny, right, long. That gives us 16 billion kilometers. And when we do a little conversion, that ends up being 3.7 billion miles of DNA in one person's body. So if we look at the length of that DNA in your body, if we would unravel all the DNA in every one of your cells and get that length of DNA, it is long enough to reach all the way from the earth where we live all the way to the sun 70 times. Not just once, but 70 times. So that's how much DNA is in your body. Yet if we crumple it up and condense it down, it would fit into the space of a single ice cube. So that kind of gives you an idea of how thin it is, right? And when we unravel it and when we look at the whole length of DNA, there is a lot of information in that DNA. And to think that every single one of your cells possesses that information, all of the information, and there's that much DNA in your body. It's amazing. Another way to think about it, we could think about it like, and now this is a little bit outdated because we don't really use encyclopedias very much anymore, but if you think of an entire set of encyclopedias, and let's say there were 46 volumes in our set of encyclopedias, right? We could think of it like every single cell has that whole set of encyclopedias in it, right? Every cell in your body contains the entire amount of information. And each of those 46 volumes could represent a chromosome, right, of a human being because we have 46 chromosomes. We could think of it like that. If we thought of it like that, like encyclopedias, 46 sets or 46 volumes in that set, and every single one of your cells has all 46 of those in it, we could take one of those out. We could take one volume out of that encyclopedia set and open it up and look at it, and it's full of information, right? In the same way, we could take one of your chromosomes out of your cells, out of the nucleus, open it up, and look at it. We could look at chromosome number 2, for example. And it would look like this. It would have a lot of information in it, okay? It happens to have 243 million base pairs, all right? And just so you're aware, this isn't a whole gene. That little stripe isn't a gene. It's actually hundreds of genes. It happens to color, take up the color in the dye when we do, when we put the chromosomes in dye, they take up color that way. We get the G banding patterns, but this isn't just one gene. It's actually hundreds of genes in there, but you could think of one gene as a little location on there, a very tiny little location, because there's a lot of genes on there. These are some of the different genes that are found on chromosome number 2. Here it's chromosome number 22. It has 49 million base pairs. It happens to be a little gene, or a little chromosome, has fewer genes on it. Still a lot. 49 million base pairs is a lot, and these are some of the different disorders and things that are linked to this chromosome. Chromosome mapping is another concept that is important in helping us know what genes are on what chromosomes and where exactly on the chromosome those different genes are located. This is just showing sort of the idea of a chromosome map for flies. And a wild-type fly is the normal, natural variety, and then mutants are the unusual, different ones. When we look at a chromosome, this is chromosome number 11 for flies, for fruit flies, we get at the location 54.5. The gene at this location. In a wild-type fly, it's the red eyes, but the mutant has a different coloring on their eyes, or vestigial wings, a different kind of wing. Here, see how they're short instead of the regular longer wings on the wild-type fly? That trait is located at 67.0. So the location, or legs, there's a different kind of leg. The trait for that is located at gene number 31, or at location 31. And some other traits as well. So we can make chromosome maps that show us the positions of different known genes. All right, keep in mind, before we really dig into the DNA structure a little bit more, keep in mind that each gene is basically typed with thousands of letters. So here we have our chromosome, and we unravel it. We unravel into this long, long, long strand of DNA. Remember how long it is. And that thin little long strip is really, remember these rungs on the ladder, and we'll look at that in a minute. But each of these represents a molecule, right? And A stands for one of them. T stands for another. G stands for another. And we'll look at that in just a minute. Review some of that. The A and the T always pair up. The G and the C always pair up. But when we unravel this, we could unravel it into all these different letters that represent the different nucleotide bases, okay? Instead of just the little colored strips, we unravel it into the letters. And don't forget that these letters aren't just random letters. They code for your phenotype. These sections code for whether you have brown eyes or attached earlobes or whatever it is, the trait that we're talking about. These sections actually represent the expression of those different phenotypes that make you who you are and give you all the different traits that you have. So they are not just random letters, but they are actually codes for different phenotypes, different genes that make up you. So we're going to look a little bit more in depth now at that structure of DNA. So here we are finally. We're going to launch into looking at the specific structure of DNA. This is review. We have done this before in the first semester, and we're going to look at it again because we need to really understand thoroughly what the structure of DNA is so that we can talk about how it replicates and what it does. So in the 1950s, DNA was confirmed as the chemical that was responsible for heredity. That's not really that long ago that people finally realized DNA was what was responsible as opposed to proteins. We have a lot of different proteins in our bodies, and people didn't know if it was proteins or if it was DNA that was responsible for heredity. And Watson and Crick, these scientists right here, these gentlemen, they actually determined the shape of the DNA. Prior to the 50s, the shape of DNA wasn't even known. And here's Rosalind Franklin. She had a significant part in this as well. And they found and discovered that this is the shape of DNA. It's this double helix structure, and it's a little lopsided. It's not a perfect spiral like a rotini pasta or something. It's a little lopsided. We'll look at that a little bit more too with the different base pairs, but it's this double helix, and let's take a look more in depth. What are these rungs? You'll probably remember, so you can pause the video if you want to write down the answers to some of these questions ahead of time, and let's look at that. Remember the backbones of this double helix? The backbones are made out of alternating sugars and phosphates. Okay, sugars and phosphates, you should remember that. The rungs on that ladder are base pairs of adenine, thymine, guanine, or cytosine. The adenine and thymine pair together always. So you always remember A and T go together, G and C go together, and that's how they always pair. Those are the nitrogen bases. That's what those are chemically. And here we just have another picture of it. We have the phosphate sugar. This is a sugar, this pentagonal structure here, phosphate, sugar. And then we have the nucleotide or nitrogen bases in the middle. G and C go together, A and T go together. And if you'll remember, these bases, the A, T, C, and G bases, are a little bit different in their structure. Some of them are double rings and some are single rings. A and G are, we call them purines. They have double ring structures. Okay, they're wider molecules. The C and the T, cytosine and thymine, they are, and also the uracil, if you'll remember from the RNA molecule, but the T and the C are pyrimidines. They have only one ring, so they're narrower. And they also have different numbers of sites for hydrogen bonding to take place. We always end up getting the A and the T pair together. The T is a single ring, and the A is a double ring. So it makes the total width about the same, and they have two hydrogen bonding pairing sites. The G and the C always bond together. The G is the double ring, and the C is the single ring. So we kind of conserve the width of our entire DNA structure by having them pair in this way. These also have different numbers of hydrogen bonding sites, and so that's why they bond together the way they do. So this complementary base pairing, that's what we call the fact that A only goes with T and G only goes with C. That complementary base pairing is what allows for really efficient, error-free replication. And that word replication just means making a copy of itself. DNA replicates, remember during interphase, we have DNA replicating so that we get a second copy of it, and it's obviously very important that it replicate as error-free as possible so we don't have any mistakes, right, in the genetic code. And we're going to talk right now about how that happens. How does DNA replicate? How does it make another copy of itself? When we start with our DNA molecule right here, these are the steps, really simple. The first step, the DNA unzips, and right here is where it has unzipped. It's unzipping right there. It splits in between those base pairs. Here we have A and T are just the next ones that are going to unzip and be split apart. Here we have the C and G that are being split apart right there. Here we have the A and the T that were split apart, and we have another A coming in to pair with that T. That's the second step, nucleotide pairing. There will be, this T is going to come in and pair with that A up here. We already have the nucleotide pairing that occurred right here, G and C, G and C. We had a C came in to pair with that G, and G came in to pair with that C. We have this new strand being formed again, and the third step in this process is that it rezips. Here's our new strand, rezipping. Here's the new strand, rezipping. And we have two identical copies. This will continue to unzip. The nucleotides will continue to pair with each of the complementary strands. The complementary strands rezip, and we will get two identical copies of DNA. One thing to note, many enzymes assist in this process. Remember, enzymes are chemicals, they're proteins that help make reactions happen, and many enzymes are going about assisting in this process. And so one more thing to note about DNA replication is that it is what we call semi-conservative. What is that word? Sounds kind of strange. When we look at our old strand, our initial strand of DNA, okay, remember it unzips. So here it unzipped, zoop, just like two sides of a zipper, right? Then we have bases that are floating about in the cytoplasm, A, T, C, and G bases that are free to bind to the correct places here to complement them, make the complementary base pairs. And we end up with our two strands of DNA. Half of each of them is old. It consists of the original old strand, and half of each of these new strands is new, consists of the new bases. And that's why we call it semi-conservative, because the old strand is being conserved, or it's conserved. It's right here. And it's being semi-conservative because it's not all the old strand, it's just half of the old strand in each of the new pieces of DNA when it's replicated. So we call it semi-conservative. So what happens if there's a mistake? If there's a mistake, we call that a mutation. One kind of mutation is the result of incorrect base pairing. Here's an example of that. We have the correct base pairing here, A and T, G and C, A and T. Whoops, that is not correct base pairing. A and G don't go together. This is a mistake. We call that a mutation. This little guy is supposed to represent an enzyme. These are little enzymes, different kinds of enzymes. They don't really wear glasses, but they actually go along the strand here and feel for making sure that we have completely parallel strands, and here it is not. See the bump there? These are not correctly paired. And so that enzyme detects that, checks for the error. We have enzymes that cut that error out. They make a cut in the DNA. We have other enzymes still that remove that incorrect section of nucleotides. And then we have other enzymes that come in and replace these nucleotides with the correct nucleotides. You can see those are replaced right there. And then enzymes that reconnect the sugar phosphate backbones. So that is one way to take care of mutations, and obviously mutations are not good for the individual. They can be very, very bad, very damaging. So enzymes are constantly going and checking this replication process and making sure that it is accurate. Advanced ideas. Here's a few ideas. Read through these. See if you would like to do some of these for extra advanced proficiency. and I look really forward to seeing what you come up with. There's a lot of other questions. If you have your own questions that you have come up with from watching this presentation on DNA and on how it replicates, please ask those questions and do some research and find the answers and come and show me. I'd love to see it. So, we'll see you in class.